Good afternoon, and welcome to this LinkedIn live interview brought to you by the Human Brain Project. My name is Philippe Ryvlin. I'm a professor of neurology and head of the Department of Clinical Neuroscience at the University Hospital in Lausanne, Switzerland. And today I have the privilege of interviewing Viktor Jirsa, who is the Director of Research at the Centre National de la Recherche Scientifique and Director of the Institut de Neurosciences des Systèmes at Aix Marseille University. Our discussion will focus on how Viktor's team is using extremely detailed brain simulation, patient-specific models, and predictive software tools to improve surgery success rate for patients with epilepsy. So I would now like to welcome Viktor to join me on screen. Hello, Viktor.
Hello, Philip. Nice to see you.
Nice to see you too. Thank you so much for being with us. So let's start with the first question. What challenge and scientific question are you addressing with this research?
Okay. I'm leading the virtual brain project. And in this context, we aim to understand what I consider one of the biggest challenges in neuroscience, and that is actually variability. And variability has been an obstacle and a barrier to progress in neuroscience, in particular inter-individual variability. When we think of clinical translation, this is a big issue. Some subjects, for instance, who suffer from an insult or who receive an insult and an impact, they never show any clinical symptoms and are resilient, and others, so they are suffering from this and develop a disease or a particular disorder. On the other hand, when you look at individual patients with the same symptomology, some react beautifully to approaches, interventions, and others do not. So this individual variability is a huge issue for clinical translation, but also in neurosciences and fundamental neurosciences. There we refer to this as neurodegeneracy, when multiple mechanisms can lead to the same type of behavior. So this, for me as a researcher, provides an obstacle in terms of identifiability. And what we are doing with our approaches, we take a structure from individual subjects, so individual structural constraints, build models, impose the individual structural constraints on a brain model, and then constraining the type of dynamics, the type of function that comes out of this. So we're actually addressing the binome of structure and function. And this is one of the oldest dichotomies that we have in biology.
We often heard about personalized medicine. I think this is really a great example of it. Will you agree? We're truly on that personalization of treatment and medicine here.
I agree. Of course, the personalization with the virtual brain tool in our hands is limited to a certain level of description, and that's a system level. But there, because of the challenges we have with structural variability, at least this is a stride forward into this direction. And for some diseases, it makes a big difference. At least those that we can describe on the structurally localized level. So personalized medicine is the direction in which we want to go. And there, the vision essentially would be to think of build a virtual brain model for every patient. Then this provides you with a platform on which you then can operate. You can act, manipulate different changes. You can personalize this a platform for a particular patient, and then you can ask hypotheses. Under these constraints, how does it change brain activity? Does it improve the situation or does it not?
Now, why did you choose epilepsy? to apply this virtual brain model?
So epilepsy, first of all, epilepsy is a very important disease. Yeah? It affects 1% of the human population. And of those, about 2/3 can be treated pharmaceutically. Whereas for the pharmacoresistant patients, it's there are much fewer options. Surgery is one, and there we need to identify the epileptogenic zone. This is a target actually for the diagnostics and later on also for the intervention, because the underlying theory is within the epileptogenic zone, you have the emergence of pathological activity, paroxysmal oscillations, that then start recruiting the rest of the brain, and through this, it starts hampering the proper functioning of the patient. So that's an issue. But you see from what I just described, it is a dynamic disease. Yeah? And epileptogenic zone, it's an expression that can be expressed in terms of location, but also in terms of connectivity. So on the level of how we work, on the system level, all the elements were in place such that we can actually treat them or handle them within the virtual brain framework. And then being in Marseille, we have a wonderful epilepsy department here with excellent clinical collaborators. It's headed by Fabrice Bartholomé, who's one of the leading researchers in this domain. So we had an opportunity to develop systematically an approach that allows us to virtualize Fabrice's patients, and we have done that systematically. That allowed us to build a battery of clinically relevant questions that we were able to address in terms of the virtual brain approach.
Now, Viktor, could you describe the model you're using in a little bit more detail? Maybe explain how they differ and what are their advantage as compared to, let's say, more classic or most standard connectivity model, and actually how you are able to simulate a digital version of a human brain for an individual patient.
Okay, so there are connectomics has become a large field since 2005, when the term connectome, the complete set of all existing connections, was coined by Wolf Kotter, that was Giulio Tononi, and Olaf Sporns. Yeah? So those three gentlemen were part of a consortium of which I was part also, the Brain Network Recovery Group, in which we investigated the brain from the perspective of a network. Today, it has become a common perspective and is shared by everyone. At that time, that was not the case. Yeah? And at that time, we were wondering to what degree can we use non-invasive imaging data to help us to better, non-invasive structural imaging data, so in particular connectivity data, to constrain function. And function in this context means, on the one hand side, brain function, but its accompaniment is brain activity. So we started discussing model approaches, and that was the origin of the virtual brain. And we recognized that, and that was in particular with Randy McIntosh and later Peter Ritter, who joined also. We recognized that all the competences and capacities that we need in order to put these elements together to build a meaningful platform that can actually build a model of a virtual brain. For this, we always needed, not a single laboratory can do this by itself. You would also always need this collaborative effort. So we decided to build this platform in which there is a pre-processing step of neuroimaging data coming from a single subject. These data are being imaged by typically non-invasive MRI and provide us with a connectome, yeah, for this individual patient, and the topography, which is essentially the geometry, cortical and subcortical areas, and we merge this together in the same space. But by that time, you have only a geometry, so it's not functional. So you have an avatar, a form. Yeah. So you need to equip this form with something that can actually generate dynamics, yeah, which is then the source, the generator for brain activity. So you need to put the math in. So you can equip the network with mathematical equations, so-called mean field equations, where the Human Brain Project in particular has contributed a lot. Colleagues of mine, such as Alain Destex, have spent a large part of their career to develop mean field equations, which actually provide informationally reduced, mathematically reduced equations that actually are derived from very, very high dimensional equations where you have the classic single neuron approach, but then you reduce it to a neural population that has only multiple degrees of freedom. This we put into this avatar, and this renders the model actually functional, because now you can actually take this specific model that is being derived from the individual's brain imaging data. You can simulate it. You can simulate it on your workstation or on a high performance cluster if you want to go in more detail. But you can simulate it and actually map from the brain region activity. You can map it on the signals that you, Philip, are using in the clinic, yeah? Functional MRI or EEG or MEG. This type of signals, let me add one more thing. These population models that are then still unconstrained, they're only constrained by the connectivity, but we can put in additional information, such as in epilepsy. What we do is we increase the epileptogenicity of a particular region. But this is not true for every region, and it's different from one patient to the other. So we need to find and build approaches where we can actually estimate this from some other criteria other than structure. And this is where then functional brain imaging data, by functional I mean temporally resolved brain imaging data that inform you about brain activity, this is where they come in. We put this in, and we use methods from machine learning, so-called Bayesian inference, to estimate these parameters and even further personalize the virtual brain model. And then you have a real individualized platform that you can actually use to either simulate the data for this patient and then check on clinical hypothesis, verify the clinical hypothesis, whether actually your hypothesis does what you think it does, verify it in this personalized brain model via simulation. Or another possibility is you use this individualized brain model as a template for parameter fitting in order to improve the quality of your estimation of the epileptogenic zone, because in epilepsy, that's, as I said, for the pharmacoresistant patients, it's actually the objective. Identify the epileptogenic zone and render it quiet, which means either epilepsy or through some other approaches that actually avoid the system or hold it back from going into epileptiform activity. So this is the entire pipeline, yeah, coming from imaging data, building a geometric form that is already personalized, equipping it with mathematics to make it functional, improving the personalization by addition of machine learning, artificial intelligence methods that are actually estimating in a very sophisticated fashion the parameters. For the experts, we typically use Monte Carlo Markov chain approaches. So we are estimating actually even more than just a set of parameters, but provides us with diagnostic value. How good are we at doing our estimation? And then the output is information that is actually meaningful to the clinician, like the localization of the epileptogenic zone or testing of clinical hypotheses.
One question regarding the output you can deliver in terms of simulation in a human being. So I understand you use multi-scale information to build the model, but then the output, what will be the best spatial resolution you could provide, taking into account the nature of the data that are available in a human being and which necessarily constrain the spatial resolution. What will you propose today with the TVB in terms of spatial resolution?
So we have started off with about 100 to 200 network nodes or brain regions. If you look at this in terms of spatial resolution, we are talking about 10 to 20 square centimeters, which is actually huge. Yeah? From a surgical perspective, it's actually good enough, because when working with our epileptologists and neurosurgeons, we are actually on the level of multiple centimeters. However, there are issues in terms of interpretation of signals, like the mapping from sources to sensors. Sensors, stereotactic EEG, so invasive EEG or EEG signals, they sample from multiple regions, and the brain has a folded structure. There may be multiple regions contributing, so we need to go at least to the millimeter range. In the virtual brain, I want to say a few more words on other approaches. So we can do this actually, and this is within the Human Brain Project. There we have accumulated data that allow us, these are post-mortem data, that allow us to inject this type of data to further constrain an individual's virtual brain on a millimeter level, or almost sub-millimeter level, and then improve the resolution, and through this the degree of detail, but also the confidence we can have on this relationship between signal, so sensor signals, and source signals. Yeah, it's called the forward solution. I emphasize this because in real world applications, in practical situations, this is one of the most contaminating factors. Yeah? So that is wonderful, but by using ex vivo data, we are losing the in vivo element. Yeah? So this personalized aspect, because they're coming from post-mortem studies. So this required approaches where we are performing, again, data fusion of ex vivo and in vivo data in order to actually get the best from these two worlds. Yeah? And again, that was possible in terms of Bayesian inference approaches. So that is what we do in the virtual brain pipeline. However, you can go vertically to even lower resolutions, spatial resolutions. Today in the Human Brain Project, we have repertoires of data coming from the human with high resolution scans. So coming from laboratories in Jülich, in Paris, in Amsterdam. These labs are working together in order to provide ex vivo data all the way down on the cellular scale. So we're talking about the micrometer scale, and that is cellular resolution of cortical tissue, but also high resolution reconstructions of fiber tracts. This is very valuable information. We are not using it directly. We are using in the virtual brain. So my colleagues that are performing very high resolution cellular simulations, like Markus Diesmann, Michele Migliore, or in Lausanne, the Blue Brain Project, they are using this type of data. We are using this type of data by constraining more the mathematics of the mean field models to obtain better and more precise representations of the reduced models that we are the so-called population models. 1 millimeter resolution for us is enough. For us in this context means we are studying the propagation and evolution of brain activity, and how it's constrained by clinically relevant features such as variability in connectome or changes in excitability. We are not on the subcellular or molecular level. However, there the benefit is that we are working together in the Human Brain Project and actually exchanging informations. And there are three major simulators. That is a neuron simulator, which provides subcellular, cellular simulations. The NEST simulator, which is using point neurons. And then there is a virtual brain where we are on the system level. But of course, you can use a co-simulation technology, not for everything, but there is communication between these simulators, so you can actually design approaches where you can actually start mixing the scales of operation. So this is fundamental neuroscience.
You mentioned different opportunities from the Human Brain Project in supporting and leveraging all your research. Are there any other aspect to mention regarding what HBP has brought to the virtual brain model?
Yes. The Human Brain Project now enters in its 10th year and approaches completion. I talked a lot about collaboration within the Human Brain Project. I cannot overemphasize this aspect of collaboration. During 10 years working together, we had to work together because we decided to work together in the Human Brain Project. So it was our choice. But this working together allowed us, we had to learn the language of the other communities. We had to understand their problems. We had to build a culture during 10 years together. And this is a value that cannot be overestimated. A shared culture, understanding what your colleague who's coming from engineering or from molecular biology or from medicine is trying to communicate to you, rather just hearing the words. The word connectivity can have so many different meanings in different domains. Yeah, this takes a long time. This culture, I'm deliberately using the expression culture. So it's because it's not just language. It's also a way of operating with each other has an enormous value. Once you share this culture, you actually make the step to integration. And you saw how I was responding to your question, Philip, in addressing different simulators, going across different scales. This is essentially integration of information of coming from heterogeneous sources. Yeah, we talked about in vivo, ex vivo. So you're adding value, you're adding real scientific value in addition to or compared to the system where you have these individual pieces of information in isolation. Yeah, and this adds scientific value. This allows you, if you embed it to existing neuroscientific knowledge, which we typically express in terms of models, you can link the data and models with the tools, the simulators, in order to generate knowledge that is not accessible otherwise. So, for instance, I often make the comparison: classic machine learning methods extract the information that is in the data. If you link it to a model, or in other words, to a causal hypothesis, you can actually extract information beyond what is in the data because you link it to existing knowledge. This culture and these workflows have been developed by the Human Brain Project. And the key deliverable of the Human Brain Project is eBrains, which is a federated digital neuroscience infrastructure, which allows us to use these tools, models, and data in an interoperable fashion. And this was key to our usage of the virtual brain in the epilepsy applications, because when I joined the Human Brain Project, we built only very generic models. In order to enable clinical translation, we needed to be able to personalize them. and we needed to develop methods to personalize them. And we chose Bayesian inference methods after having tried many others. You can choose naive optimization methods where you maximize the correlation of activity between areas or between experimental and simulated signals. But this will not get you anywhere, because we talked about variability and neurodegeneracy. There are too many challenges. Yeah, so you need to use these sophisticated methods of what is called actually causal inference in order to be able to have a reliable and form of personalization that comes along with diagnostics, allowing you to know how bad or how good your estimation is. For this, you need computational resources, you need mathematicians, you need to understand the data, you need these workflows that are now available in eBrains, that are now interoperable or being pushed towards interoperation. It's still in development. We are pushing it forward. So this is, without that, this would not have been possible. Yeah? Without that, we would still be at generic virtual brain models. And this is not translatable into the clinics.
Well, let's translate it to the clinics now, because what is very exciting for a clinician like myself involved in epilepsy surgery is this huge clinical trial, randomized clinical trial that you are been developing in Marseille with Fabrice Bartholomé to test whether using the virtual brain will have an impact on surgical outcome and providing high-level evidence. It's really a unique trial. Can you tell us where we stand now in this ongoing trial? Maybe how many patients have been recruited, and when do you think the trial will be finalized to provide some findings?
Okay. So the clinical trial, it's called EPINOV, and it has for the target, for the primary objective, to evaluate the value of the virtual brain in the clinical decision making for the identification of the epileptogenic zone. It started in 2019, summer 2019. We it was supposed to finish actually summer this year, but like everything in our lives through the pandemic, where everything got slowed down. Right now, we have an inclusion of almost 300 patients. Target is 400 patients in order to arrive at a sufficient statistical significance to make actually a statement, whether to be able to make a statement whether the virtual brain has actually improved the surgery success rate. Of these, so we have included about 300, almost 300 patients. Of those, we have two arms. Yeah, one arm which is VEP, so virtual epileptic patient. This is how we call the workflow, VEP as an acronym. So we have a VEP positive branch and a VEP negative branch. In this trial, there are 13 clinical reference centers in France that are involved. Half of the patients this is a VEP positive branch. Half of the patients is receiving this information of virtualization, this added value of estimating the epileptogenic zone through the virtual brain. The other half is not. The way it works is actually the reference centers, they are a normal clinical routine. They collect the data. They provide all the data that we need for virtualization. They are anonymized, centralized in Lyon, being sent to Marseille. We are performing the virtualization, the estimation of the epileptogenic zone, and we are sending the results back in form of a clinical report. And the clinical report is then being used in the individual clinical staff meetings for the decision making. And as I said, in the VEP negative, branch, this is not being considered, and in the BP positive branch, this is being considered. We should arrive to have a full patient inclusion by May, June 2023. This is when we will stop inclusion. And then we will start with the data analysis. Until now, we don't have any right to interfere with the trial. A patient is being considered to be seizure-free if he or she has not had any seizure during 1 year after the surgery. So we have to wait even 1 year after the surgery, after the stop of the inclusion, to have all results in place. Yeah, but the first analyses, the first results should become available end of 2023, I hope.
Thank you, Viktor. My last question to you today is about potential other application in the future in epilepsy or in other neurological disorder. Do you want to say a brief words about those potential using the TBB?
Yeah. Actually, at the moment, one of the very hot topics for TBB, but also in many applications with my colleagues and other laboratories, that's actually stimulation. There are different forms of brain stimulation. You can do this invasively or non-invasively. You can do it in a diagnostic form, so you can use it for diagnostic purposes, or you can use it for therapeutic purposes. There are different forms of stimulation. In epilepsy, for instance, under certain stimulation protocols, you can actually reduce the degree of epileptogenicity in epilepsy patients. So this is a very exciting area of research, and in my opinion, there's the biggest promise to provide results in the next 10 years, even potentially with new technologies such as temporal interference, to be able to render diagnostic and therapeutic stimulation completely non-invasive, because you can actually go into profound brain areas and focalize on individual regions. But you need guidance. You need to understand how to stimulate. You need to understand how to do this for this particular patient and not just generic. So this is where the virtual brain can, first of all, provide guidance for navigation in a personalized brain template, but then we can also optimize the stimulation protocols, so we can render the tissue active such that we can show responses and the tissue will be active in the model as a function of its connectivity or as a function of its hypothesized epileptogenicity. But this is not just epilepsy. Stimulation translates also to other applications. Yeah? We have neurodegenerative disorders, Parkinson's disease, or pharmacoresistant treatment of pharmacoresistant depression, for instance. It opens up a new domain that under TBB guidance we can now explore. So I'm, and many of my colleagues are actually very excited about this potential application of TBB for clinical, in the clinical domain.
Thank you very much, Viktor. It has been a pleasure to share the screen with you today, and I learned a lot, and I'm sure all the audience is here. Our next LinkedIn live interview will take place on September 28th at 12 CET, and will feature Georg Conrad, an associate professor in the Division of Computational Science and Technology at the KTH Royal Institute of Technology, and Yannick Morel, researcher in the Faculty of Psychology and Neuroscience at Maastricht University. They will be discussing their work on safer interaction between humans and robots. We hope to see you then. Thank you for joining me, and I wish you a great afternoon.